Well, thanks very much, Andy. As Andy pointed out, I have this sort of relationship between the University of Guelph and Brock University and CCOVI, and spend a fair bit of time traveling back and forth because recently my research has centered around viticulture and in particular precision viticulture. And in my earlier research work, which involved field crops such as corn and soybeans, I became interested in using remote sensing, and in particular airborne remote sensing from low altitude, so that we could do some weed identification and some spatially variable spraying and those sorts of applications. But in the last 4 or 5 years, I've moved into the area of viticulture. It's an industry that is locally very important, obviously. It's also an industry that is characterized by a lot of variability. And consequently, looking at precision viticulture, as I've indicated here, is a particular interest in the Niagara region. And in the background here, I have a airborne image. This is a color infrared image, and this is just a vineyard shot from the Niagara on the Lake Virgil area. Anyway, to get into the precision viticulture, why is there an interest now in precision viticulture? And really, if you look at the Niagara region, then, of course, everybody knows the terrain is highly variable, and we have variation in things such as topography, soil, microclimate. You could also consider the terroir term, because the variability that we see gives rise to perceptible variability or changes in wine that's produced from some of these areas. In the Niagara region, it has become quite clear that different areas in the same vineyard block may in fact differ quite a bit in terms of nutrient availability, canopy vigor, fruit quality, and that sort of thing. So the idea is, in precision viticulture, how do we manage that variability and what do we do with it? And of course, one of the things that we can try to do is to homogenize it and try to make things more uniform, even out the production across these variable areas. Or another approach might be to look at adapting to that variability and essentially capitalizing on the small changes that we might see within these zones, particularly in terms of then segregating fruit at harvest perhaps and coming up with some unique products or reserve wines from these different areas. Of course, like most things under the sun, this is not new. This has been going on for a while, and some of the earliest work happened in places such as Australia, New Zealand, in California, Oregon now, and moving into BC. And initially, this idea behind remote sensing grew out of the problem with phylloxera. Now, that's not really an issue in this part of the world, of course, because phylloxera is native to this area, and of course, we look at rootstocks that are tolerant. But in places such as California and New Zealand and Australia, then there was a concern with the decline that was caused as a result of the phylloxera root louse. So initially, the remote sensing was started as a tool to monitor the spread of phylloxera and try to find infested versus non-infested areas. Down in Napa in California, Lee Johnson, sort of in the early 90s, late 80s, early 90s, working with NASA, looked at remote sensing, and his focus at that point was this attempt to keep track of the phylloxera infestation. However, he found that when he was collecting this remote sensing data, it had other uses as well. Could be used for things such as crop scouting. Many things obviously popped out from the imagery that was being collected, harvest planning, and so forth. Commercial services began in California and the northwestern United States around about 1999 or 2000, something like that. Well, when we talk about remote sensing, of course in Canada, remote sensing is quite a mature industry because with our emphasis on natural resources such as forestry and minerals and that sort of thing, remote sensing has seen a lot of research, a lot of development because of our wealth in those particular areas. But if we think about forestry, or if we then move into viticulture, the idea is that in remote sensing we're looking at a couple of things. First, we're looking at the green vegetation, and in most of this remote sensing work we're looking at ambient, or using the sun as the radiation source. So we're looking at reflectance that's coming back from the canopy. And of course you know that we have molecules such as chlorophyll that absorbs in certain wave bands, 420, 490 nanometers, for example. We know that vegetation has a green peak in the area around 550 nanometers on the spectrum, the electromagnetic spectrum. We also know that chlorophyll has an absorbance trough in the area of 660 to 680 nanometers, and so forth. We also see a near-infrared plateau in green vegetation where we get a lot of reflectance in the near-infrared, or that part of the spectrum that's just beyond our human vision. And we also see some, for example, water trough features in the spectrum as well. So we characterize the spectrum of the vegetation, and if we look at leaves that are under some type of stress, and of course phylloxera was one of those stressors, then what happens is you see a shift or a change in the spectrum, the reflected spectrum that are coming from the leaves. And typically this shows up in the green, yellow, orange part of the spectrum, and also in the red area. And we see a lower reflectance that shows up in the near-infrared. Frequently what happens is when we look at remote sensing, we're looking at taking different parts of the spectrum and combining them into the form of an image, which we can then interpret. So one of the ways of combining some of these spectral bands is in the form of indices. And there are many different types of indices that are currently used, but some of the most common are what are called the Normalized Difference Vegetation Index, or NDVI indices. And these essentially combine either the red and the infrared part of the spectrum, or the green and the infrared part of the spectrum. Andy mentioned that I spent some time in Australia. While I was there, I was involved in a couple of research projects. One was looking in Victoria, and we had some vineyards where there were some phylloxera infestation, and we had a lot of vines there with the original rootstock. What we have here is a typical sort of a spectral reflectance characteristic that comes from green vegetation. So as I mentioned before, down here, this is the visible part of the spectrum down here. So this bump is the green peak, and then we have the red trough down here where we have a lot of chlorophyll absorbance. And then we get the red edge where suddenly we have a great increase in the reflectance. So we see on the near infrared part of the spectrum, fairly high return. About 50% of the incident radiation is reflected back again. Now in this particular slide, I've gone quite a bit out into the mid-infrared part of the spectrum as well. This is well beyond the human range of vision, and we see a couple of features here at around 1480 and around 1950 nanometers. These features are due to water, the influence of water. So there is information in the spectral reflectance that has water status associated with it, albeit the systems that we're using don't really capitalize on it because we cannot sense up into that mid-infrared part of the spectrum. But if you look at these two different curves, I have the red curve and the blue curve. The red curve is an infested population. This is, of course, is an average from many, many different samples that we took. But basically, spectral differences show up down here in the visible part of the spectrum. So you see that the infested vines have a higher peak and also a higher reflectance in that sort of orange-red part of the spectrum. We see some differences up here in the near-infrared and then farther down here in the mid. What does this mean? Well, if we're to combine information that we gather on those vines then, we can partition them, and in this case what I've done is I've used an NDVI green ratio, which is the combination of near-infrared at 740 nanometers and green at 550 nanometers into a normalized index, which has a range from -1 to +1 and a mean of 0. But in this particular case, vegetative return is always going to have a positive NDVI. And when we look at plotting these, then we have the non-infested plot up here, where the NDVI is above about 0.55, and the ones that are sick plot down here below. So one could conceivably partition the different areas that we're sensing into two zones, one which is probably infested and one which is probably healthy. Well, in addition to the spectral information, though, these remote sensing images have a lot more information. One of the basic things that people realize when they first look at these, and of course you all realize it yourself because whenever you go up in an airplane, you look out the window and you look down at the ground and you see patterns that you never realized existed as long as you were earthbound. So you see different colors, particularly in the spring. You see different soil color variations relating to moisture. You see different vegetative type of variations as well. Well, these show that there's all sorts of underlying variability that we can see from the vegetation. And this variability may have to do with soil, moisture, fertility, and disease, and so forth. But in addition to that, from a single image, we can get a lot of spatial information. But if we have a sequence of images, what we can do then is by combining them together, we can look for changes or trends that are taking place over time. So this can be, for example, based on weather effects, drought, very prevalent last year in this area, could be disease spread, and so forth. So the spatial information that's in these images is important as well. If we then take these images that we create and georeference them, and what I mean by that is that the individual picture elements or pixels in the image are tied to a specific location on the Earth's surface, which of course relates to global positioning coordinates and that sort of thing. So once we georeference the image, then we have all sorts of other tools at our disposal. We can determine areas, we can determine specific locations, we can measure distances, all sorts of things that we can do. Most of you are quite familiar with remote sensing in one form or another because most of you have used Google Earth and some of these net-based packages that are out there right now. And that type of remote sensing is based on principally satellite images, although there are some high-altitude aircraft images in their ensemble as well. But our approach is slightly different. One of the problems with satellite imaging is, for one thing, you're up above any clouds that may be sort of intervening between you and what you want to look at. The other thing is that the atmosphere absorbs particularly a blue part of the spectrum. And the other problem with satellites is they're usually overhead at specific times, and it may not be when you want them to be there. So our approach was to take a much lower cost approach. And essentially what we do is we have a system that we can fly from small fixed wing aircraft. What we have done is taken actually a door from a crashed Cessna, and we rebuilt it with a bubble that sticks out from the side. And the idea is that we can remove the co-pilot seat from inside the aircraft. We can mount our camera system on the seat rails so that it looks straight down, and then we take off with our plane and we fly around, typically at around 3,500 feet above the ground because we want to get relatively small pixels on the ground. And this is our system. Basically, the reason that we chose the aircraft is that it's inexpensive, it's flexible because we can fly weather permitting whenever we want, and we can go exactly where we want and we can come back to that spot over and over again. Now, the sensor that we're using actually is a multi-band CMOS camera, and the way that we capture these images, and this is a view over Thorold, essentially what we end up with is a number of different images that are in a grayscale. So they're monochromatic, and what we do then is we combine them together. So in this case, we can combine red, green, and blue, overlay them, superimpose them, and we get the regular color image that you're familiar with, because the tristimulus colors, red, green, and blue, give rise to our perception of color. But in terms of infrared, which you can't see, we also, one of the cameras captures in the infrared part of the spectrum. And these images, if you've done any near-infrared photography, you realize it looks quite different. And that is because you get this very high vegetation in return. So trees, for example, appear very white in near-infrared. So when we combine the near-infrared and the red and the green information, then we get what's called a color-infrared composite, another type of a remote sensing image. So now in this case, you can see a lot of the features on the ground show up. Some of them are emphasized more than others. Vegetation is really emphasized, as you can see. The colors look a little bit bizarre because we're looking at a combination that doesn't give rise to a normal color sort of image. However, there's lots of information in those images that allows us to extract information from the scene. Here, for example, is an image that's taken. It's a color infrared composite, and it's over a Riesling block, and this was taken at the end of August in 2007, a very dry year, as you know. You see this red area down through here. Well, this is right in the vineyard. This shows that at this time we have quite an active canopy. We're getting quite a bit of return from the leaves, and of course at this point that's what we want because we're past veraison, but we still have a lot going on as far as the plant is concerned. However, up in here you can see there's an absence of red color, therefore there's an absence of active canopy at that time. And of course down here this is the bush, and it's quite active, although there are a fair number of spots in there where it's showing some moisture stress problems as well. If we look at a different vineyard, these patterns start to become fairly obvious when you look for them. Here's a particular vineyard. We have a Riesling block here. We have Chardonnay over here. This image was taken in June 29th, 2007, before a lot of the real drought issues started to be noticeable. But you'll notice that there's sort of a meandering pattern that goes down through here. It's not very obvious at the end of June, but if we look over at the other side, this is the same field imaged at the end of August. You'll notice quite a difference now. You'll see that there are some red areas here, and this meandering sort of red pattern goes down through. Well, what's that telling us? Well, it says that the vines in this area are still reflecting a lot of infrared, so they're still fairly happy. The vines in these lighter areas here are not reflecting as much infrared, so they're showing more signs of stress. And of course, what's happening here? Well, when I talk to the owners of this, they say, oh yeah, there used to be an old water course that went down through there, and that's probably what we're looking at. What's causing the variation? Could be a number of things. It could be soil type, it could be soil moisture, could be organic matter, it could be a number of things. Once we have the image, then we can combine information in the image to get more information coming out of it. One of the things that we can do, which I've done in this image, is I've classified the image so that I can interpret what I'm looking at. So I've taken a classification where now all of these that you're looking at look kind of weird because they're false color. They're not what you're used to seeing, but of course with the computer-generated colors we can create whatever combinations we want. What I've chosen to do is use red to show active vegetation, broadleaf vegetation, so therefore you get the bush shows up in red. You also can see the grape rows now coming out because they show up as vertical red lines. Parts of the image that are not vegetated show up in black, and other types of vegetation which aren't the sort of broadleaf characteristic response, and in this case, in this particular image, it's mostly floor vegetation, and it's mostly sort of browned off because it's at the end of the season. Then by combining the spectral information that's in the image, then we can see other patterns start to come out. So for example, here you see this broad swath of green that goes down on a diagonal from, well, this is northeast, this is southwest down here. And we see another one up here. Well, what we've done here is taken the color infrared image and created a normalized difference vegetative index, and it highlights vigorous canopy. So in this case, I've chosen my colors so that yellow is poor vigor, bright green is high vigor. Therefore, the bush comes up quite green, and that means that this is vigorous vegetation. This along here is shadow because we took the image a little bit after noon. Well, I mentioned earlier that not only do we get the spatial and the spectral information, but we can also get temporal information from our images. So what I've done here is I've combined two images, one from the 20th of July, one from August the 14th, and I've done a subtract on them so that I see the difference in this normalized difference vegetative index. Or in other words, how much has it changed during that period of time. So that shows me where the canopy is increasing, and I've shown it in green. So where you see green, you're seeing a positive change in the canopy NDVI. Where you have blue areas, these are negative changes. Or in other words, the canopy is going backwards. It's not reflecting as much in August the 14th as it was back in July. Again, probably related to moisture problems. Well, that brings us to the 30 Bench Winemakers Project. And the images that I've been showing you, the last few, of course, you recognize this dog leg in all of those images. It's a very particularly useful feature to us when we're up in the airplane because that helps us find that vineyard up on the Beamsville Bench. And you would be surprised when you're flying over the bench areas how similar all of the vineyards look. So it's quite easy to lose your target. Anyway, what we decided to do, and this was after Dr. Richard Smart, the flying doctor from Australia, was over and actually did a talk here at CCOVI a couple of years ago. And Daryl Brooker up at 30 Bench and Richard contacted me and they said, we think we got a good candidate site for you to do some of your remote sensing work. And they were right. This, basically this block here, all the way around this dog leg is all Riesling. So they had a large block of Riesling. What they wanted to do was to divide the block into smaller management zones. And initially what we did was we looked at some vigor analysis from the vegetation remote sensing in 2005. So we divided these things up into different zones, and that's why you see these different colors superimposed on the image. The idea was then we would follow these zones, we would harvest the fruit, and we would vinify it separately, and we would see what happens if there are perceptible differences between and among these different zones. So we want to determine the variability of fruit, we want to determine the effect of that on the wine that's being produced, and so forth. Of course, if we're buying into this zone management idea for precision viticulture, if these zones aren't stable across years, or in other words, if these zones move around in the vineyard every year, then they're not much use to us because we can't predict the fall from what's happening in the spring of this particular year, for example. We need to know that they're stable. So we went in the first flights were back in 2005, as I mentioned, and we identified these zones, and we identified them based on the canopy vigor as it was at the end of 2005. It wasn't perfect. We had a few constraints that were imposed upon us. Daryl said, well, I don't want any oddball-shaped management zones because I have to harvest this stuff, so we had to come up with essentially rectangular type of boundaries. That didn't necessarily overlay on the areas that we were defining. And he also said, well, they have to be of a particular size. If they're too small, they're no good to me. You have to make them large enough to make it viable to separate them at harvest. So we came up with these 6 different zones in that Riesling at 30 Bench. Now, I mentioned earlier, one of the other advantages of this remote sensing imagery is that once it's georeferenced, there's a lot more information now in behind the image that you can make use of. So here's that same vineyard block, only what I've done now. This is georeferenced, so that means that north is up. If you want to click on the corner of the building over here, for example, the imagery will reveal to you the GPS coordinate of that building, and you can take your little handheld GPS and you can go and you can walk up to it, and lo and behold, yes, that's where it is. Of course, nothing mysterious about that because the building doesn't tend to move around very much. However, when we get out into the vineyard, we have these little yellow X's that you see here. These indicate the GPS location of an individual vine, or what we call sentinel vines. And the idea is that year after year, we want to go back to these same vines, we want to harvest the fruit from them, and we want to determine how they're performing based on their earlier data that we collected. So those actually are where they occur on the image and also on the ground. So for example, if I wanted to find out what was happening with that particular vine, I could get the GPS location coordinates from the image, and then I could walk out and find that precise vine. Now, we tend to use belt and suspenders here too, so we also put flagging tape around all of the vines so we can easily find them on the ground as well. But the point behind the GPS is that if we had an automated harvester, if we had say a fertilizer spreader, maybe a manure spreader, we wanted to apply manure to this particular site, then we might use the locations of these vines to tell us whether we needed more or less, or any other type of variable input that we might want to apply. The other information, of course, as I've done here, I've drawn a boundary around this area. This is my LV1 management zone, and out pops the size of it. So it's just over 7,400 square meters, basically. Same for all of these. So we can now go in, and if we want to, for example, if I wanted to find out the area inside that little ellipse there, because it maybe was a diseased area or something, I could find that out very conveniently. I can also immediately determine lengths, because all of these pixels that make up this image know where they're located on the Earth's surface. So these sentinel vines are established in order for us to track the evolution of these different sites. So we flag them and we georeference them, and we go back to the same vines year after year. We're into the third year of the project now. We collect canopy information, soil information, fruit characteristic data. Also, we group the fruit from the sentinel vines together and do small batch vinification so that we can see what's happening in terms of, in this case, we're grouping by water stress. In addition to that, then the larger management zones are harvested separately, and that fruit is tracked as well through the winery. So the winery keeps the zone batches separate through their process. We make use of a lot of graduate student labor. This is Luca Bessat. He was one of our French students visiting last summer. So what we did for this project, we needed to get ground data so we could evaluate our imagery data. So we got soil moisture from portable TDR, vine water status from pressure bomb measurements, for example. We did individual vine leaf reflectance spectra. Then we did the normal suite of harvest data, and sensory and chemical data was collected as well. In order to make the connection between the airborne remote sensing imagery, where we're getting spectral reflectance, and the health and stress on the vine, we would like to fine-tune the operation a little bit, because there are many different parts of the spectrum we could conceivably use. We need to know which parts of the spectrum have the most useful information for us. Because this 4-band camera system that we have, we can change the spectral bands at will. We can move, for example, if we don't want to have the blue band, then we can use that camera to capture a band in some other part of the spectrum. So we look at the actual leaf reflectance in order to determine which parts of the actual spectrum are responding to different types of things. 2006 was the first year that we got really a complete suite of data from the site. Guess what? 2006, very cool, very wet. If we look at the average moisture, and this is the average of a number of TDR measurements taken over the season, what we see, these little color cartoons as some of my colleagues like to refer to them as, however, you could look at that, and the human eye and the human brain, being very good organs for determining positions and patterns, you can immediately see that there is some patternation to this thing. It's not just a random bunch of amoeba on this slide here. But if we look at the, look here, this is the range of soil moisture. extremely high in 2006. We got some very wet areas down in here. This is the northwest corner of the block. We have some drier areas over in here, but even the driest areas are still in the area of 16 to 20% moisture, so they're not really dry, not in 2006 anyway. But what we see is this pattern, sort of a diagonal pattern, which pretty much mimics the pattern that we were seeing from the remote sensing image that I gave you with the NDVI on it. Again, no big mystery there. However, if we then overlay on the same blocks the yield data, then we see some interesting things develop. Here we're having low yield. The low yields down here are in blue. This is in kilograms per vine. So blue up through yellow towards red. Well, here are low yielding areas up in here. Well, these just happen to correspond with the wet areas that we were getting. Not exactly, but pretty close. But then we see the same thing happening. We see this diagonal zone running down through here where the yield is in sort of the reasonable area. I should mention to you, Linda Tremblay did her undergraduate thesis here last year, and this data was data that Linda actually processed and extracted from what we collected in the field. So if we look at other parameters that were collected at harvest as well, for example, if we look at plotting sugar or bricks at harvest, here we have the higher sugar areas. Well, in 2006, these corresponded to the low yield and the water stress, but in this case, in 2006, the water stress because it was too wet. Down in here, where we had the lower moisture, the bricks levels are in that sort of 18 to 20 range. Linda also looked at chemical constituents, and she looked at, well, a number of different measurements, but I've grouped together the total amount of terpenes because these are important in Riesling. Then we can see similar types of patterns, not quite as obvious, but you still get this diagonal running down through here from northeast to southwest. We have low levels here, low levels down here, and we have higher levels up here. Now up in here, this was the block that we originally in 2005 had categorized as a low vigor block. So it appears that in this year at least, we were able to see some benefit of, or offsetting benefit to that low vigor area, which gave us low yield, in that the monoterpene level was quite a bit different. If we look at all four of these cartoons together, you can see that there is in fact quite a marked pattern that occurs. Every one of them has this sort of northeast to southwest diagonal arrangement. It's not perfectly preserved in every one, but obviously there's some consistency. There's some connection between what we were seeing from the images and what we were collecting from the vines in the field. Well, then, of course, 2007 comes along, and what do we have? Almost the total opposite situation. Much hotter than average, much drier than average. If we look at plotting the average soil moisture, you'll notice for one, the scale is much different now. It's only going up to 20%, half of what we were seeing in the previous year. Anyway, here we have a very dry area. Here we can't call it a damp area, but at least it has more soil moisture available than most of the rest of the vineyard. But again, it's arranged in this diagonal sort of pattern. If we look at the yield in 2007, the pattern is somewhat preserved. The yield levels are quite a bit different, but the high yield shows up in here now. So where previously this had been our low vigor area because we had too much water, we had low yield in 2006. In 2007, well, is it related to the soil? Yes, probably it is, because when we were harvesting in 2006 in that area, we were walking through water in many places. But we have higher yield in a dry year, and our lower yields now show up over in here. So the pattern seems to be repeated, although the outcomes are somewhat different. So we have the same sort of triangle or diagonal patterns that emerge, and these between soil moisture and yield. Now we don't have the rest of the chemical data at this point. We have another student, Mathieu, that's working on this, and that's going to be overlaid relatively soon. The first question is, are these zones stable? Well, yeah, they seem to be. At least in this vineyard, you get this same sort of diagonal pattern that appears in all 3 years of the study. And these zones are evident in the aerial images. So we can see them from the remote sensing. We can see the effects when we look at plotting the actual harvest data. At this particular site, probably due to soil type, topography, these are linked, of course. This is on the bench area, so you have lots of slope and lots of microterrain as well. The effects of the zones, of course, change from a wet year to a dry year. So now, most of you that are into viticulture and know your vineyards very well know where these zones are already in your own vineyard, and you know what influence they're going to have from year to year. But you may not be able to very accurately delineate these things as we can from our remote sensing image. So you probably know where they are, you know how they affect the operation, but you may not have a really good handle on precisely how big and where they are located. So how can we manage this? Well, a combination, I think, of the ground data, which is obviously important, but the remote sensing information, which we can supply, presumably on a relatively regular basis, so year by year, and that might help us make some decisions then in terms of what we're going to do in terms of management. What else are we going to do? Well, in 2005, when we did the zoning, basically in the main block of the vineyard, these were the zones that we came up with. Well, on the basis of the 2007 and 2006 data, though, we probably would go back. These dotted lines indicate the original boundaries of the blocks. We would probably go back and redraw those to correspond more closely with the actual variations that we're seeing. Now, would the vineyard manager be happy with this? Well, probably not, because this block may not be too bad, but when you have little irregular blocks like this that you have to harvest and manage, it's going to create a problem. Now, if you're hand harvesting, it's less of a problem, but nevertheless, we would like these things to be nice, predictable, regular shapes, but in fact, they're probably not. So we can cut some corners, and that's in fact what we did. We trimmed this, and we put this block in with this block. What else is coming up? Well, in addition to what we have been doing with this color infrared multiband imagery, we're also interested in getting into the longer wavelengths. You remember I showed the plot of the spectral reflectance of vegetation, and we had those two trough areas that indicated water status. Well, if we get into longer wave and all the way out into the thermal part of the spectrum, in the mid-infrared we can get water information. In the long wave, we can get thermal information. Thermal information is important because it gives us temperature variations that we see that are due to the canopy status. So if the canopy is actively transpiring, it's keeping itself cool, as opposed to canopy that's not able to keep up with the water demand, the temperature is going to be different. So we look for changes in surface temperature variation, and we look at heating and cooling patterns, and from that information, then maybe we can get some more information about what's happening in the vineyard. On the left, we have a color infrared image of a particular vineyard down by the Niagara on the Lake Lakeshore. And when we look at this was done at the end of August. This particular site has overhead irrigation, has pretty good canopy coverage, as you can see, but there are some spots here where it's less than in other areas over here. This is all one clone, one variety in here. Over on the right-hand side, what we have is a thermal image. We did a small project where we got a thermal camera and we did repeated flights over the same area and we captured a thermal image. So one thing you'll notice here is that we have this is calibrated in terms of temperature in degrees C. So these different colors correspond to different temperatures. So the lower temperature, around 28 degrees C, that corresponds to these purpley blue areas. Well, that happens to be quite happy canopy, so the temperature is cooler. We did a sequence where we started at dawn, well, actually a little after dawn, because they don't like us flying around at 3,500 feet when it's dark. So we did a sequence where, and here I've combined 4 of these, where we've got a scene that was taken in the morning, just after dawn, at noon, in the afternoon, and then at dusk in the evening. And you'll notice a couple of things. First, the temperature changes, the temperature scales change here quite a bit. On this one, the highest temperature is 27 degrees. Well, at noon, the highest temperature is quite a bit higher, 44. And then towards evening, it starts to cool down again. So what we're looking for now are patterns that emerge in not just the spatial variability or the distribution of these things, but how it changes with time as well. You'll notice here, for example, in the afternoon, that patch, which has not very much canopy intercepting the radiation, is very hot, about 45 degrees, because what we're seeing mostly at that point is soil. We're not seeing a lot of canopy. So the canopy is not able to, or the surface is not able to keep the temperature down because it was very dry and it was not able to sort of offset the solar influx coming from the sun. So that's one of the things that we want to do, is we want to expand this remote sensing more into the thermal area. Back in the 70s, and in fact, the establishment of the zones in the Niagara region were based on thermal imaging that was done by Bob Ryerson and other people from the University of Waterloo back in the 1970s. What else do we want to do? Well, the other thing that we want to do is we want to take this remote sensing idea, combine it with other information, specifically weather information, so real-time information on rainfall, humidity, temperature, things that are important for viticulture and disease and that sort of thing. So we have another research project going on, which is sort of a parallel operation. And what we're trying to do is, working with a number of collaborators across the peninsula, is come up with or work towards a system of collecting this remote sensing information and then passing it on to the growers and the winemakers so they can do their own interpretation. So we need a system. Right now, I have this little plane that I rent, and I can go up and I can image parts of different vineyards, but I can't do it on a full-time and on a large scale. We have to have a whole system in place that would allow us to do that. And then that geospatial information can be extracted, combined with weather data, and used for management decision-making. I mentioned here we have some of the partners here. We've got Kim Geomatics, Environmental Geosolutions, Weather Innovations, and Agricor. This is being principally sponsored by the Ontario Centres of Excellence through the ETEC program. Anyway, I presented quite a bit of information, I guess, in a fairly short period of time. I certainly would be remiss if I didn't mention at least some, and certainly not all of the people that have been involved in this. Principally here at Brock, Andy Reynolds has been invaluable just through the facilities that are available, his knowledge and access to the students and everything else. Daryl Brooker at Hillebrand, 30 bench. And then we have a list of students here. Unfortunately, I couldn't mention everybody, but all of these people played a very important role in collecting data for us. But of course, without money from the Ontario Centres of Excellence and NSERC, we wouldn't be able to do very much at all. Obviously, the industry collaborators as well, 30 bench winemakers, Brock CCOVI, Weather Innovations, Aviation International, and Lakeview Harvesters, to name a handful. And that is really all that I wanted to present this morning. This is obviously a work in progress. We have 2 years of data. The intention is to extend this at least for another year and maybe longer, as long as interests stay focused on this. And hopefully, yes, in a few years we'll see remote sensing as a vineyard management tool that's widely available, not just in the Niagara area, but across Canada. I know we have some people here from Prince Edward County. We've had interest from the Okanagan and Lake Erie North Shore and so forth. So that's where I better stop. And if you have any questions, by all means, I'll try to answer them. If I don't know the answers to them, then Andy's here. I'll put him on the spot. Thanks. John.
First vineyard at 30 Bench, is that tile green?
Yes.
What district?
I can't tell you that for sure. It was not laid out on a really regular grid spacing, though.
And second question, can you do that thermal imaging from satellite yet, or just limited to what you're doing with airplane?
Well, no, you can do it from satellites because, in fact, if you look at satellite imagery available from Weather Canada and that sort of thing, you can get thermal imaging. But, of course, what you won't see is what's happening on the ground in this detailed sense.
I don't know, but thinking of like from Google, if you get detailed temperatures.
You can get on a large scale, and when I say a large scale, probably 10-meter pixels or something like that. So you're not going to see too much detail of the actual vineyard at that scale. But the other difficulty, of course, is if there's a cloud or something like that, you're not going to get thermal information. Not very reliable thermal information anyway. Of course, if it's cloudy, you probably don't have to worry so much. Yes, Kim?
Well, I'd like to just continue on with one of your closing statements. Now that you're developing this information, what is your vision for the future? How do you see this being applied in precision viticulture?
Well, the first task is to convince other people that it's actually useful. So from a research perspective, it's interesting, it's somewhat novel, it's useful for our purposes, but unless it's useful on a larger scale to producers, wineries, it's just another one of those things that, you know, it's a curiosity, but it's not going to go any farther. So what we're working towards is trying to make a system that actually presents information that is useful to the vineyard managers and at an affordable rate. So, you know, nobody's interested in paying whatever, $200 an acre or something like that to get imagery done. But if we can get it done for $3 or $5 an acre or something like that, and we have coverage over a large geographic area, then it might become viable as an industry.
And are you referring specifically to pest management and fertilization applications and those sorts of things?
Once you have the information about the variability within your site, then there are lots of different things you can use that information for. Planning operations would be one. One of the things that we had hoped to do in 2007 was to look at the mildews and see what might happen in terms of them. So we actually did a weekly scouting in our collaborator vineyards. But in 2007, we didn't have a mildew problem really to worry about. But what we were hoping to do was to make a connection between the change in canopy volume over time and how that translates into unprotected canopy, so that if you're into a regular spraying sort of regime, you know where the important bits are, and you know where there are some areas where they're still important. But if you're going to put your money on getting out there first, this maybe is where you want to go. So I think there is a connection between, and the bottom line is we really don't know the answer. Right now we've been doing planning in terms of harvest scheduling, maybe take this part of the block off first and leave it because the Brix is low in the rest of the block, and then we'll come back and harvest it later. One of our collaborators, in fact, did that in 2007. They went in and they told their pickers, you know, harvest these areas, leave these areas, and they were able to see quite a dramatic change in their bricks at harvest. So many of these ideas. In most of these situations, the really useful information is going to come from the growers because they're the people that know. And so what we need to do is listen to them and let them tell us what would be useful. Yeah.
Was there any sensory work done on the segregated fruit from the 30 bench?
Yes.
Zones?
Yes, but I don't have that data. Yeah.
You used it on stationary pests like blotts or on leaves. Is there any application for pests that are on the move, like leaf hoppers or malb?
Yes. Well, I don't know about melb because you're just not going to see anything at the resolution that we're looking at. You're not going to see anything happening. In the case of leafhopper, you could see some changes in the canopy reflectance. And there again, it would probably not be from a snapshot that you would see it, but it would be from a sequence of images to see, oh, I see this region that's different, and then next week the region is moved or it's gotten larger, or maybe not next week, maybe 2 days later or something like that. So if we were into that sort of a diagnostic situation, we would have to be able to put more flights up, you know, on demand. We're also considering that if we were to be able to come up with a system that could monitor water stress, then we could use that for irrigation scheduling. So if we did sort of like last year, if we had started at maybe the end of July and gone up every week or 2 weeks or something like that, then we might have